Hello and welcome to this Nature Research custom media webcast titled CRISPR Goes Viral, Biology and Applications of a Bacterial Immune System. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by GenScript. CRISPR-Cas is an adaptive immune system of prokaryotes. Apart from protecting its host against invading viruses, alternative biological functions of CRISPR-Cas have also been revealed, leading to transformative applications in research and medicine. This webcast will detail CRISPR discoveries and inventions and their ongoing impacts. We'll begin by hearing from one of CRISPR's pioneering scientists, Dr. John van der Oost of Wageningen University and Research in the Netherlands. It was his work on unraveling the mechanism of CRISPR-based immunity in bacteria that paved the way for developing CRISPR-mediated genome editing. We'll then move on to a question and answer session with you, the audience. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them at the end of the session today. And now, over to Dr. van der Oost.
Thank you very much for this kind introduction, and it's a great pleasure to talk about CRISPR, about the biology and all the applications that have been developed over the years. Before I start my presentation, I want to give a statement on conflict of interest. So I am involved in 3 different companies, Entrance Technologies, Scope Biosciences, and Hudson River Biotechnology, mainly as a scientific advisor. And my name is as inventor on top of several CRISPR-related patent applications. Okay, so what I aim to do today is talk about the biology behind the whole CRISPR system, and then I want to specifically talk about 2 defense strategies that have been unraveled over the years, and also from these 2 strategies, different applications have been derived. So let's start with the with the basics. So the diversity of CRISPR, which is a result of the host virus arms race. So what you see here is a cartoon of a viral particle that recognizes a suitable host organism, in this case a bacterial cell. So after recognizing and interacting with the cell wall, it injects its genetic material, in this case DNA, which is being replicated and transcribed. Let me get a pointer. Hang on. So it's being replicated, and then it's transcribed and translated into proteins. The proteins make up the viral capsids that at some stage package the newly formed genetic material, and then complete viral particles are being formed. And they break in many cases they break the cell wall of the host, and the viral particles are released, and then the cycle can start all over again until some resistant host organisms are encountered, and that's part of the arms race. And some of these systems have been known already for more than 50 years. Basically, they have been discovered during the 1960s. And we can now discriminate a wide variety of different antivirus systems. And they operate at different levels of the viral proliferation cycle. So it all starts with the interaction of the cell. So basically, there are systems that can prevent the infection, thereby blocking the entry. Then there are intracellular systems that can detect an infection through the presence of viral nucleic acids. And these systems they prevent the replication of these nucleic acids just by cleaving the DNA or the RNA. And then last but not least, and that's something that becomes more and more general, that's systems to prevent, in fact, the spreading of the viral particles, and that can be done by generating a dormant state of the host organism, but it can even lead to suicide of that host organism. And by committing suicide, basically the cell sacrifices itself to save the rest of the population of related bacteria. Okay, let's now move to the CRISPR-Cas system. So CRISPR stands for clustered regularly interspaced short palindromic repeats, but I will come back to the details in a minute. But these CRISPR systems, it's good to remember that there are many different CRISPR-Cas systems, and they are all a consequence of the ongoing arms race between the viral invaders and the host organisms that usually carry these CRISPR systems. And if we look at these CRISPR systems, then we can see that basically there are two strategies that I already mentioned. So the first strategy is that there is specific recognition and cleavage of the viral DNA or viral transcripts, but also and sometimes also some of the bacterial viruses also have an RNA genome, so also that can be recognized and targeted. But apart from that, also mechanisms have become clear over the recent years that some of these CRISPR systems induce a dormant state of the host or even suicide of the host. Also, what I already mentioned is that this arms race is really very important. That also gives rise to a lot of diversity. So we can say that this arms race between the parasites and the host organisms is a major driver of evolution, and that also can be illustrated by this cartoon that we recently published in a review. So if you want to know more details, I advise you to have a look at that one. So what you see here basically is different stages of that arms race. So this is the very first one, when a viral particle can use a certain bacterium as a host and use that to infect and to proliferate, and at the end also to kill the cell. But then some of these organisms, as I already indicated, they kind of evolved defense systems such as CRISPR-Cas, and that can then neutralize an invasion, meaning that not the phage but the bacterium is the winner. But then also, as a next step in the arms race, the viral particles have been found that evolved a next step, and that's anti-CRISPR systems. So that kind of interferes with the activity of the CRISPR system at different levels. And then, of course, the phage again is the winner. And recently, a next step has been found in which the host organisms produce anti-CRISPR systems. So this clearly indicates that there is a lot of selection pressure to come up with new inventions in the course of this biological arms race. Okay, also then about I should mention the diversity of the CRISPR systems, and that's huge. And basically, a couple of years ago, we decided to come up with a new classification in which two classes are in fact discriminated, and class 1 includes all the CRISPR-Cas systems that are using multi-subunit complexes called cascade complexes, and I will come back to that in a minute, associated with Cas nucleases. And two of these class 1 systems have been well characterized. That's the type 1 system that next to its cascade complex, it uses a Cas3 nuclease. And here you see the cascade complex. Each different color depicts a different subunit. Here you see a type 3 system, and this particular system has a cascade-like complex, and it is associated with a Cas9 nuclease. Then in class 2, maybe you heard more about those systems, because class 2 are the systems that are very popular for genome editing applications. In this case, it's a single protein that does the job. So it has the nuclease activity, but also several other activities which are distributed over different domains. And probably the best known system is the Cas9 system. But also Cas12 is, it has been discovered only a couple of years ago, but it's also a very interesting system for genome editing applications, as we will see. And then, last but not least, there is the type 6 system, of which the key player is Cas13. I will come back to all these systems, and I will every now and then I will kind of summarize where we are. So hopefully that creates some clarity in this very highly diverse CRISPR-Cas mechanism. So this is the first conclusion slide. So what we now learned is that CRISPR-Cas is a highly diverse defense system that you can find in prokaryotes, both in bacteria and in archaea. And that both DNA and RNA can be targeted in a specific way, and that's basically the first strategy, and that's to kill the parasite, so to kill the invading virus. There is also a second strategy, as I indicated, and that's dormancy or even suicide of the cell. And by doing that, it kind of sacrifices itself to save the remainder of the population. And as you will see, that linked to these different strategies, there are different applications that have been developed over the years. Okay, so let's move to the first strategy then. So that's the specific targeting. So here you see again part of a bacterial cell with the CRISPR array. So the CRISPR array is a cluster of repeats and spacers, and the repeats are all similar, spacers are all different, as indicated by the different colors. And a typical size of a repeat is something like 30 base pairs, and the same is true for spacers, but they come in different flavors if you look at the different systems. And next to these CRISPR arrays are the CRISPR-associated genes, so the Cas genes that code for the Cas proteins. Well, if such a cell that possesses such a CRISPR-Cas system is infected by a virus, then in most cases the virus will be the winner, because because in most cases there is, or in many cases, there will be no matching spacer in the CRISPR array, and that means that it cannot directly neutralize such an invasion. But in a subfraction of the infections, and that's, for instance, that has been demonstrated in the case of inactive virus mutants, then the immunity can be gained through the CRISPR-Cas system. And how does that work? Okay, well, we can divide the CRISPR-Cas mechanism in three different steps. And the very first step is the acquisition of new spacers. So basically, what happens here is that there is recognition of the viral DNA, and then selection, and I will come back to that, selection of certain fragments for new spacers, and those fragments are referred to as protospacers. Okay, so then there is selection, and then what happens is that the CRISPR array is opened at one site, so then double-strand breaks occur, then duplication of the repeat part is being generated, and then the new spacer is being integrated. So then we basically have an adapted CRISPR array, and basically you could consider the CRISPR array as the memory of this adaptive immune system. Okay, then of course the memory has to be expressed, so in the form of RNA. So this goes via general transcription, and then processing gives rise to mature RNA fragments that are called the guides of the system. So the CRISPR-RNA guides, and they are bound to effector complexes, effector Cas proteins or Cas complexes. Then the final step of the mechanism is the interference, so the target interference. So recognition of sequences that match, so that base pair with the guide RNA. And then an important feature is the fact that there are these so-called PAM motifs. So a PAM is a protospacer adjacent motif, and that is very important because that allows for self-non-self discrimination. And by doing so, it avoids autoimmunity. And this PAM sequence is as is indicated by the name, so it's adjacent to the region that is being recognized by the spacer, because the spacer is derived from the protospacer. So and it's very important at the level of target interference. So only when that PAM sequence is there, then cleavage will occur. And also that means that to make the system even more efficient, that also at the level of selection of new spacers, only sequences are selected that are next to such a PAM motif. But the part that is integrated does not have such a motif. So in the actual CRISPR array, there these PAM motifs are not present. And so these regions are not cleaved by the system because the PAM is active, and only the region that does have a PAM, so the invading sequence, is being targeted. So as I said, this avoids the autoimmunity problem. Okay, then back to 2008. That was the time that we in Wageningen selected the E. coli CRISPR system to get some first biochemical insights in how this system works. And what we did here is we transplanted different sets of genes from the E. coli K12 strain to the E. coli BL21 strain, and together with a designed CRISPR in which several spacers were integrated that had matching sequences with the one of the E. coli phages, phage lambda. And so together at some stage we found the right combination, and this in fact was the eureka moment where we understood that these 6 genes together with this designed CRISPR could be used to provide immunity to E. coli against this specific lambda phage, the specific virus. Okay, so what did we learn? We learned that the CRISPR indeed is being transcribed as one long precursor CRISPR RNA, which is then being processed by one of the Cas proteins that is part of this complex that I already mentioned, the Cascade complex, standing for CRISPR-associated complex for antiviral defense. One of the subunits is responsible for the maturation of the guides. Then the Cascade complex or some other proteins, some other subunits of the Cascade complex, they are responsible for binding of the CRISPR RNA guide. And then in the case of the Cascade system, this nuclease Cas9 is responsible for targeting DNA, so to cause severe damage, as we will see in a minute, severe damage of the viral DNA that is being recognized by the CRISPR RNA guide. So it's RNA-guided DNA interference. That is at least what we concluded after analysis of this E. coli system. Another important thing is that we were able to transplant the CRISPR-Cas system from one bacterium to another one, so to another host. And also we could, by making or by using a designed CRISPR, we were able to target specifically different DNA targets at the same time. So in this case, we designed spacers to make guides for targeting 4 different genes of this particular E. coli virus. So this was the first example of multiplex DNA targeting. So not only did we find the important features of the molecular basis of the CRISPR-Cas mechanism, we also provided some starting point for genome editing. This movie kind of summarizes where we are now. So here you see the assembly of the Cascade complex with the CRISPR RNA molecule with its hairpin structure, its palindromic structure over here. So that's kind of the anchor to the Cascade complex. And then the stage is ready for screening, so surveillance of the target. So here you saw that there was indeed a PAM sequence, but not a matching DNA sequence. But in this case, you will see that there is, next to the PAM, there is indeed the protospacer from which this guide has been derived. So here you get a complete R-loop formation, meaning that there is double-stranded DNA formation, so a base pairing between the guide and the target. And then this Cas9 nuclease appears at the scene. And I guess you can appreciate from this movie that this is a perfect antiviral system, because it's causing major damage to the viral DNA. And so this is well, it all makes a lot of sense that this is a very useful system to specifically recognize and neutralize viral particles. So what did we learn? All CRISPR systems use RNA guides that are derived from the CRISPR. So we call them CRISPR RNAs. The Cas3 Cascade system that I just described to you, but also the Cas9 and the Cas12 system. And maybe you remember that Cas12 and Cas9, they are belonging to class 2. I will come back to that in a minute. Whereas the Cascade system is a class 1 system. Two systems that I did not talk about in a lot of detail yet are, the Cascade-like system, the type 3 system that I mentioned in the beginning, and that's targeting RNA, and also a class 2 system that targets RNA has been revealed a couple of years ago, and that's called Cas13. So a lot of diversity, and I can imagine that for some of you this is a bit of overwhelming, but I will come back to this at several stages in the presentation, so I hope it will be clear at the end. What about the applications of this strategy? So the specific targeting, how can that be applied? Okay, well, we initially were thinking of applications of the Cascade system, but there are some drawbacks there. As I indicated, it's a perfect system, as I mentioned, to damage viral DNA, but it's maybe not the perfect system to do precise genome editing. But still, several applications have been developed. The very first one is to make use of this devastating activity, and that's to introduce large deletions up to 100 kilobase pairs or more even. So huge deletions, they can be made using this system. The other applications of Cascade are in the absence of the Cas3 nuclease, and that's just to make use of the binding capacity, for instance, to block a promoter region, hence to influence transcriptional or the transcription of a particular gene. So just by designing the guide, you can block a certain promoter. Then a very interesting natural variation on the CRISPR theme was found that basically the Cascade system was hijacked by a transposon. So a transposon is also just like a virus, a mobile genetic element, a selfish DNA basically, but also that system kind of took from probably from a bacterial system. It took the cascade part and in this way repurposed it. And this also can be used to kind of introduce in bacteria. So far, it didn't work out yet in eukaryotes, but I know people are working on that. But so far in bacteria, this system has been used to introduce 10 kb fragments, for instance. Then the last application that I mentioned here is an application of Cascade in which the Cas3 part has been replaced by the FokI restriction enzyme, and this has been demonstrated. So we did that in collaboration with Sam Sternberg and Peter Cameron of Caribou, the company, and it took many years. It was very complicated, but at the end we got it to work to also in human cells do genome editing. But that said, this is not the ideal system. The ideal systems are class 2 systems. So first of all, that is the Cas9 system, of which it was demonstrated that the cleavage, if you compare that to the cleavage by the Cascade system or by the Cas3, nuclease, in the case of Cas9, that's really what you want, because here you get double strand breaks in a very clean way. So no exonuclease activity, just endonuclease activity. So you get blunt and double strand breaks very specifically. Another class 2 system is Cas12, and that is very different from the Cas9 system. You can see that if you compare in red the different RNA molecules, the different guide molecules. In the case of Cas9, there are 2, so it's the CRISPR-RNA together with the tracer. In the case of Cas12, there is only a single CRISPR-RNA. Also, the PAM is at the other side, and also the cleavage is pretty much different. In this case, there are 2 active site or 2 nuclease domains involved. In the case of Cas12, both strands are being cut by a single nuclease domain. Okay, another big advantage that I already indicated is that the Cascade system uses these Cascade-like complexes consisting of sometimes 5 different subunits in an uneven stoichiometry, so relatively complex. And then you also need this additional nuclease. In the case of Cas9 and Cas12, it's an all-in-one system. And that's why people often refer to that as the Swiss Army knife. Okay, so these systems are well suited for precise genome editing, and so the only thing you have to do is to design a guide at the level of the DNA of the chromosomes of the organism that you want to add. Then you have to introduce the system in those cells, and then when all goes right, you get cleavage. That's basically where the role of the CRISPR nucleases in genome editing stop, because then it's the actual editing is then carried out by the repair systems of the cell that you're working with. So that's either a non-homologous repair system that's very useful for inactivating genes, because a quick and dirty repair system, the non-homologous end joining system, is being used. For precise editing, you want to make use of a different repair system, the homology directed repair system. That for that you can add next to the Cas protein and the specific guide, you can also add a repair template, and that then is being used by the by the cellular repair system to get precision engineering. So then, apart from these precise editing, also novel approaches have been developed over the years by many different groups, and I listed the most important ones, or I hope I included all the important ones over here. So over the years, many different systems have been developed, and I'm only going to mention them very briefly. So it's the base editing and the prime editing systems that really have a bright future. I would say they are developed at the Broad Institute by the group of David Liu. And then these error-prone editing systems. So that's basically based on a nuclease of Cas9 that is fused to an error-prone DNA polymerase. So very locally, a nick translation occurs, and thereby mutations are randomly introduced. Then, just like the Cascade system, also one of the Cas12 systems has been hijacked by a transposon. So again, that can be used for guided transposition and delivery of major DNA fragments up to 10 kb and maybe even longer. And then again, in analogy with the Cascade system, also transcriptional regulation can be done by dead versions of the enzyme. So you just bind it to block a promoter region, or you can fuse the system to activator domains that enhance the recruitment of the RNA polymerase, thereby enhancing the expression. Okay, to summarize this part, so the specific targeting is a general strategy used by all CRISPR-Cas systems to neutralize viral infections, and many different tools have been developed based on Cas9, also of a little bit less of Cas12, so far at least. Then also the Cas13 is a system that edits RNA. I mentioned that, and also there. Or this system has been used to effect edit specific RNAs for specific purposes. Of course, that is what is being brought forward always as an advantage, is that you don't generate permanent changes. Of course, there are also some drawbacks. Then about the Cascade system, well, I mentioned for major deletions you should still use the Cas3, but for specific editing you should use this FokI restriction enzyme that is synthetically fused to the Cascade system. That can also be used as an alternative for genome editing. Then the other RNA targeting system, so the Cascade-like system that targets RNA, no real applications have been published so far, but in principle you can also use it for silencing gene expression. And as you will see, there are also some other interesting applications of this particular system that I will move to now. Oh, and that's so let's now move. So apart from the specific targeting, the different strategy is the collateral damage, and that is what we move on to now. So to compare with the Cascade system that targets DNA with this Cas3 nuclease, this type 3 system, so also belonging to class 1, so also it has this Cascade-like system. Instead of Cas3, it has a Cas10 nuclease. And as I already indicated, it's not targeting DNA, but it's targeting RNA. And the way it does that is, again, it's causing major damage to the RNA. And basically the active sites for that are residing in the subunits that make up the backbone of this cascade complex. But there is more to it, and that is causing, so this is all specific cleavage. So this part of the also of this type 3 system is specific targeting of RNA, but there is more to it, and that's a very interesting system that has been revealed by the Marafini and Jinek groups, and in parallel by the Sixnis group. And that's that there is an interesting signal transduction pathway, and that is that part of the Cas10 subunit has the potential to form these cyclic RNA molecules. So in detail, they look like this. So ATP is converted to a ring of 4 or 5 or 6, and that depends on what Cas10 we are looking at. But small rings of RNA are being formed, and those rings are being used as a messenger molecule to activate, in this case, ribonuclease, meaning that also it's being activated. So this then gives rise to collateral damage of any RNA. And not only that, also the Cas10 subunit is able to have a collateral activity, and that's DNA's activity. So collateral cleavage of any DNA. So this is only specific. These two are aspecific. What does that tell us about the biology of the system? Well, the specific cleavage obviously is to neutralize the viral infection. But when that does not work efficiently enough, then a second stage can occur, and that's that the RNA of the host is being degraded, and that then initially could give rise to a dormant state, and then when that's not even sufficient, then also the host DNA can be degraded, and that then could result in suicide of the system. Okay, so about this non-specific cleavage, so we saw that it happens in the type 3 system. There are also some reports in the literature that also the type 1 systems, so connected to Cascade and Cas3, that they also have some mechanism that resembles collateral damage, but details are not known yet. A lot of details are known about two class 2 systems. First of all, is the Cas13 system that was kind of discovered by the team of Feng Zhang and Eugene Koonin. When they characterized this new type of CRISPR system, they found that not only specific targeting occurs again of RNA, but also collateral cleavage of any RNA. And then, last but not least, also the Cas12 system has been demonstrated by the group of Jennifer Doudna, not only to cleave specifically the target DNA, as was already found a couple of years before, but also that this system can execute collateral cleavage of any, in this case, single-stranded DNA. Okay, so what did we learn? We learned that this non-specific but target-induced cleavage activity. So there should be a target, but that then induces this non-specific cleavage. That effect is a general strategy in many of the CRISPR systems. So far, not in Cas9. So that's basically the only exception so far. And that the target-induced non-specific cleavage of host RNA and DNA can be executed by the Cas10 system, as I showed in quite some detail. You see here summary of that. But also Cas12 can target host DNA, and Cas13 can target host RNA. So the question is, can we use this for application? And of course, the answer is yes. And here again, I summarize or I use this little bit more detailed slide that I took from a paper of Jennifer Doudna, in which you can see that the Cas12 system is being activated by a specific DNA, but it can be, and that's interesting, it can be both single-stranded DNA or double-stranded DNA or single-stranded DNA, and both of which give rise to activation of the nuclease domain, which is then positioned such that it can attack any single-stranded DNA that is in the vicinity. And this also, as said, this also works in the case of the Cas13 system in a slightly different way. But on the basis of this, two diagnostic tools have been developed. So the DNA targeting system based on Cas12a is called Detector by the Berkeley team, and the people at the Broad Institute of the group of Feng Zhang doped the name, or came up with the name Sherlock, and for their Cas13 system. And when they use these systems, so the activation of the system with a specific sequence, if they combine that with specific fluorescent DNA or RNA sequences, you can have a very sensitive readout. Now, many different applications have been put forward, and maybe in the times of the corona pandemic, these systems have played a big role in the detection of infections. In fact, and some of these systems have even recently been approved by the FDA. So this is really bringing the systems to the market. A recent development, and I'm almost there, a recent development is based on the type 3 system, where a combination of a pre-amplification step with LAMP at elevated temperatures, together with a system from moderately thermophilic organisms, can be used also for the same purpose. And the good thing of this type 3 system is that you kind of have an amplifier based on the production of these cyclic RNA molecules. So also these systems, they have several advantages, and one of which is that they are they can be operated at elevated temperatures, which has advantages for the whole detection system, and also the sensitivity is comparable to these class 2 systems. So to wrap up, the Cas12 and the Cas13 systems have initially been developed for very innovative diagnostic tools. But the last kid on the block here is the type 3 system. So it has been developed both by the team of Blake Wiedenheft in Montana and by our team in Wageningen. So what did we learn today? So CRISPR-Cas is a highly diverse adaptive immune system of prokaryotes, and that CRISPR-guided, so CRISPR-RNA-guided nucleases can be used to specifically target DNA and RNA sequences in the biology of the invading virus. But of course, they also provided a basis for beautiful applications. And it's great that as we speak, clinical trials are ongoing, both from natural and synthetic Cas nucleases to cure human genetic disease. And apart from the genome editing applications, also tools have been developed based on the non-specific cleavage. So, which is also very important in the biology of the whole system. But again, applications have been developed. So novel diagnostic tools have been developed with the systems that I have mentioned in detail. So to wrap up, this is what you should remember. There are many different CRISPR systems, so the class 1 systems and the class 2 systems. There are two different strategies by which they act in biology, but also how they can be applied for beautiful applications. And with that, I want to finish by showing the people that did the work in my group, but also all the great collaborations that we had over the years with people in Boston, in Bozeman, in Bethesda. That's the group of Eugene Koonin, but also in Berkeley with Jennifer Doudna, David Taylor, and with Blake when he was working with Jennifer, but also several labs in the Netherlands. And I cannot be complete, not even with all the people that I list here. But I want to thank all these people, also the people that worked with me in the past, for all these beautiful results that gave rise to this wonderful story, I would say. And I want to thank you for your attention.
It is now time for our question and answer session. To ask a question, please type it in where it says type your questions here, and then press submit. So thank you very much, John. If you would switch to your camera now, I will bring you on the screen.
Can you see me?
Not yet. If you stop sharing, we should have you back on the screen in a moment. Brilliant. Thank you so much for that, John. We have a lot of questions coming in, so I'm just going to start with right away with this one. As a genome editing tool, how does the CRISPR complex segregate out from the genome after editing?
Well, that's a good question. It's also a very important question. I do not know all the details, and I think that differs a lot per CRISPR system. But what I do know is that it's not hampering the repair proteins to appear at the scene of the generated DNA break. So some systems hang on to some of the breaks, but there are plenty of examples that in many different cell types these systems work very well in combination with these repair machineries. So I don't think it's a deal breaker.
Okay, thank you. That's good to know. Our next question asks, how long a minimum and how long a maximum of nucleotides can be introduced by this system?
Well, the minimum could be 0. I mean, you could basically change one base pair, so you replace one for the other. So that doesn't give a net result then. And of course, you can. I mean, the deletions that I mentioned, for instance, with the Cascade-Cas3 system, they are up to or over 100 kb. I think even a number of I've read a number of 200 kb in human chromosomes, but also in bacterial cells. I mean, numbers of close to 100 kb have been reported, and then. So what was that the complete question? Or
yes, that's perfect. That's introductions and deletions. So yeah. Thank you very much. Our next question is on the bacteria. So about 50% of the bacteria have a CRISPR-Cas system, but what about the others that don't?
Well, the others, they have different systems. So there is really a lot of diversity in different defense systems. And it's especially, I mean, it's already, I should mention two names, the group of Eugene Koonin and the group of Rotem Sorek. And basically, Rotem Sorek was building on some insights that were published by or at least to some extent by Eugene Koonin. And the two of them, they really came up with a lot of new defense systems. So what we now know, or the general picture now, is that each particular organism has a set of maybe 10 different defense systems, many of which, at least in bacteria, are kind of concentrated on the chromosomes of these bacteria. And that because of the arms race, some systems cease to be effective because sometimes, for instance, of these anti-CRISPR systems or anti-Argonaut systems or anti-restriction modification systems, and then they either have to come up with a new invention or they let go of that system. And there is a lot of horizontal gene transfer of different different systems that we know are involved in the defense. So long story short, that each organism has a different set of defense systems.
Okay, thank you very much. Our next question is twofold, and it asks, how is it possible for the CRISPR-Cas system to work in eukaryotic cells, especially with chromatin packing? And the second question, which is for genome editing, some labs use cloning to express Cas enzymes versus directly delivering the protein. And what are the advantages of this? Perhaps take the first question first, and then we'll come to the second one.
Okay. The first question was about the chromatin. Indeed, that can explain why some genes in some organisms at certain stages of the cell cycle are more difficult to target than others. That indeed is something that it could be a problem. People. I'm a microbiologist myself, so we have limited experience with this. But other people told me that in most cases, if they try different guides to target a particular gene, that in most cases it works reasonably well or very good. So indeed, it could be a problem to kind of lower the efficiency, but overall, I think that. In most cases, it should work, especially if you are dealing with dividing cells. Then about the delivery, either as DNA, maybe even make stable clones that produce or that express the editing system versus the delivery as protein. I think the delivery as protein has advantages because it's kind of a temporal state. So also if you think about GMOs, well, basically what you do is you do not, if you deliver as an RNP complex, so the Cas protein with its guide, then it should not be considered GMO. So that has advantages. Of course, also the other system may have advantages if you want to repeat over and over again, editing with certain different guides to target different genes. So that could be a reason for deciding that you want to generate a clone. And of course, it's easier. You don't have to produce the protein, but then of course for hopefully delivery in the future to cure patients, for instance, with to enable gene therapy for certain complicated genetic diseases. There we probably have to rely on delivery through viral factors, and then of course you you do that, or you package these viral particles, and then delivery goes via DNA again. So there are many different strategies. All have pros and cons, and it all depends on your application, what you should choose for.
Thank you very much. Our next question is on PAM. How does the host cell get rid of the PAM sequences in its genome when it integrates into the guide sequence?
Okay, well, as the name suggests, so PAM stands for protospacer adjacent motif. So the part that's being integrated is the protospacer, and when it's integrated in the CRISPR, it's called spacer. But the PAM motif is adjacent to that protospacer, so that is not being integrated, so it does not end up in the CRISPR. Hence, it is not activating the system, so the system will not attack itself. So no autoimmune problem.
Okay, thank you for clarifying that. The next question is about multiplex DNA targeting. Is it necessary to end the crRNA construct, as in repeat spacer 1, repeat spacer 2, repeat, with a repeat?
Yes, because that's a good question. So for multiplexing, it has been described also with Cas9, but it works more efficiently with Cas12, because Cas12. I use the term Swiss Army knife. Well, Cas12 could be considered as the thicker version of the Swiss Army knife, because it also has the potential to cleave its own, or to process its own precursor CRISPR-RNA. So if you then deliver the gene or the protein together with synthetic designed CRISPR array, then the Cas protein itself will be responsible for cleaving the system, and that's why you need to repeat, because in the repeat, not only is that hairpin structure that I pointed out, so the hairpin structure of the mature guide at some stage acts as a kind of an anchor, so to stably bind it to the Cas nuclease, but also it contains, in the case of Cas12, but also in the case of Cascade, it contains the cleavage site for the processing. So without the repeat part, you wouldn't have processing, and you wouldn't have stable interaction with the protein. So that's not a good idea.
Thank you very much. Our next question asks about your type 3 system, and it asks if step 2 and 3 of the type 3 system happen in an order, or if they happen at the same time.
I think they happen at the same time, because it's a good question, and I know that people are looking into this carefully. And but what I do know, and also from the work that we did in our lab, that was published last year, that when you have a specific interaction of the CRISPR-RNA with its RNA target, that then gives rise to activation of this Cas12 nuclease, the Cas10 nuclease domain of the type 3 system. And this Cas10 nuclease domain is kind of bifunctional, and so that's already giving the answer, I guess, that you have activation of the system, making it a system that can be involved in the polymerization of these ATP molecules, giving these cyclic RNA molecules, and also activation of the DNA cleavage domain. Some systems, though, they lack this DNase domain, but they still do have the domain that is involved in this signal transduction system, so the generation of these molecules. But to answer the question, I think they are both. When the target really binds stably over the whole region, and especially close to where the Cas10 system is located in the complex. When there is proper binding at that level, then you get activation of the whole Cas10 system. That's my current view.
Okay, thank you very much. Our next question asks if you can discuss further the difference between transcription control performed by CRISPR with Cas9-12 and by the Cascade complex without Cas3. And the question asks whether they could differ in duration of inhibition and therefore in their applications.
Yeah, that's a good question. I don't know if people really compared it. I think both systems work pretty well. So the class 2 systems based on Cas9 and Cas12 and the Cascade system that also works well. I mean, at some stage, I mean, it of course the interaction heavily relies on the number of base pairs that are being formed. In the case of Cas9 and Cas12, that's something like between 17 and 20 base pairs. In the case of Cascade, it can be slightly more. It can be up to 30 base pairs. So based on that, you could predict that maybe the stability of the Cascade system is better, but then I would be careful with a statement like that. So I think you should just do, I don't know that people really compared in a similar system these systems next to each other, and that's I think you should do to give a final answer there.
Okay, so that's quite interesting. Some opportunities ahead for further research. Our next question then, it asks about viral P2A sequences, which are sometimes used in the CRISPR-Cas system to insert 2 genes in a single vector.
Right.
Can you elaborate on the mechanism of action in this?
Yes, well, I'm certainly not a specialist, but I can say something. I mean, the interesting thing is when the people at Caribou, I mentioned that when they kind of repurposed the Cascade Cas3 system for, the Cascade FokI system, so the synthetic variant, when they expressed that in human cells, well, they tried different things. They did it all on separate plasmids, so altogether 6, which was pretty amazing that they got that to work. But at some stage, they did it on a 2 plasmid system, and on one plasmid, or maybe even a single, or it should also be possible even to do it on a single plasmid system. But anyway, one or 2 plasmids, and on one of these plasmids, they fused all the Cascade subunits together. And they were separated by these P2A sequences, and I seem to remember that at the level of translation, that there basically the cuts are being generated. So there the breaks occur that allow for all the subunits to form this the functional Cascade complex, which to be honest, when they proposed this experiment, or when they mentioned that they were going to try, I thought, I thought it's never going to work, but they got it to work, and that's really amazing. So it is an interesting system to be used in also in eukaryotic cells, if it makes sense to deliver as fused genes.
Thank you very much. Our next question is about the viral delivery method of the CRISPR-Cas9 system to cells. Does the virulence go away as you expand the cell line?
So the virulence of So for instance, they use the AAV system, and they so basically they use the viral capsid of the adeno virus. They use the capsid, and they package the kind of a plasmid molecule that codes for the nuclease and the guide, and then when it's delivered into a eukaryotic cell, an infected cell, then there it will be expressed. I don't really know what is meant with the virulence. I mean, at some stage, so there is no issue with the virulence of the virus, because that part of the DNA has certainly been removed, so it's safe to use it. I don't know if the person who asked this question refers to the fact that we at some stage observed also some toxicity of Cas9 itself. But if that's the case, then well, it's hard to figure that out now. But that person can always send me an email, and then we can talk about that in more detail.
Thank you very much. And I think probably our last question for today, what would be the best system to generate a specific knock-in to a genetic site in a cell, in your view?
Yeah, that's a difficult question. I mean, of course, I am biased in that the systems that I worked on or that I was involved in the discovery, I see them as my babies. So I hope that they will do a better job than the others. But that's of course nonsense. But I think there is no easy answer. I think that for some systems, Cas9, for instance, works much better. For other systems, Cas12 could be better. For instance, if you look at what enzyme is being used in human applications, it's mainly Cas9, but that's also for historical reasons, because it was developed to the level that you could use it for editing earlier. But I think that also in human editing, Cas12 is catching up to some extent, at least. If you look at plants, most people use the Cas12 systems. And sometimes people, you can read in the literature many different things, and they sometimes mention toxicity of Cas12 or toxicity of Cas9. I mean, I think it depends on what cells you are dealing with, and then it's trial and error to see what works best.
Okay, thank you very much. That is where we are going to need to finish for today. We are aware that there are a number of questions that we didn't manage to get to, and we will get back to you offline after this broadcast finishes using the details that you've provided. I'd like to thank Dr. John van der Oost very much for being with us today and for answering your questions. I would also like to thank the webcast sponsor, GenScript, and of course, you, the audience, for taking the time to be with us today. Remember, you can watch this again at any time on demand at nature.com/webcasts. Thanks for watching, and I hope you'll join us again soon.